Okay, so I'm going to keep this really brief because you have two articles to read this week. So this week you are going to have the opportunity to hear a presentation from Chris Depner on sleep and circadian physiology. Just a little bit about his background. So he has a bachelor's from Western Washington in cell biology, went to Montana State studying nutrition and exercise science, then transferred to Oregon State and studied human nutrition, and then UC Boulder for his postdoc in sleep and circadian physiology. So, really impressive background, lots of education, definitely passionate about this area of study, and can speak to the nutritional piece and how nutrition is impacting sleep and sleep is impacting nutrition, et cetera. So it's a pretty cool, pretty interesting combination of education. In his bio, he talks about two primary goals for his research. One is elucidating the mechanisms of cardiometabolic disease associated with short sleep duration and circadian misalignment. And the other is developing countermeasures that integrate sleep and circadian physiology, physical activity, and nutrition in order to enhance overall health, reduce the economic and metabolic burdens of cardiometabolic disease. So the underlying portions are sort of what I hope that you'll hone in on. So we've already talked a lot about cardiometabolic disease this semester, as well as in your case study, you had to design a program to talk about how physical activity sort of interrupts these pieces. Chris is going to come in and talk about sleep and how that interacts with a lot of the chronic diseases that we've covered this semester, or Dr. Depner, I should say. So a couple of things that he has worked on or is working on. Again, I'm just going to focus on sort of these bolded, underlined pieces, but feel free to read, to stop the video and read through things if you'd like. So he talks about weekend catch-up sleep, and you'll see that referenced in one of the articles that you read this week. Talks a lot about circadian misalignment and timing of food intake. Identifies biomarkers related to sleep. I believe his postdoc or his PhD student Misha does a lot of that work. And then he has been funded by the NIH and has an R01. So I wanted to bring these up because we did talk about them earlier on in the semester. The NIH is one of the primary funders for research and is sort of responsible for producing scientific knowledge, whereas the CDC is more concerned with implementation. Dr. Depner does a bit of both in terms of the implementation side as well as the sort of producing knowledge side of things. And then an R01 refers to an institution like the University of Utah that can qualify for the highest level of grant funding from the NIH. So there is a one grant that's larger than that, I believe. I think it's a U1 grant, which means that you are integrating resources from multiple different universities, but the R01 is the largest grant available from the NIH, indicating that your institution has enough resources to carry out the in-depth level of analysis that you've proposed in your research study. Okay, so some article-specific content. So you have two articles this week. One is a sort of a narrative review. So mostly we've looked at systematic reviews so far, which combines the statistics of different papers into sort of a summary statistical paper. A narrative review is just indicative of the fact that you've looked at a lot of different research papers in the field related to this topic, and you're sort of summarizing the content for the reader. So it's a pretty intense article, but also really informative. So I hope that you'll enjoy it. It definitely covers a lot of useful material in terms of sleep. And then the other one is called Morning Circadian Misalignment During Short Sleep Duration. So this is the first article that is primary research that you will have read this semester. So in epidemiology and in public health, we tend to focus more on really large studies. This study was carried out, I believe, in 16 individuals, 8 females, 8 males. So really different than what you've read in the past. It's good. It will expose you to some of the individual studies that make up the systematic reviews that we've focused on this semester. So a couple of terms that will be helpful to understand. First is Life's Essential 8. One of the narrative review, that first article talks about it. The American Heart Association included sleep as a component of it. Used to be Life's Essential 7. It's now Life's Essential 8 because sleep was then included. So you can see the other components of Life's Essential 8 on there. So diet, physical activity, blood sugar, weight, blood cholesterol, blood pressure, and nicotine exposure. And then you can see sleep. So it's interesting, right? Our course has sort of touched a bit on diet, not a ton, definitely a lot on physical activity. We've talked about how that influences blood sugar, weight a little bit, blood cholesterol for sure, blood pressure for sure, right? And now Chris is going to come in and talk to you about sleep. So we are covering these areas of life that significantly impact health. And hopefully that gives you some context for how to sort of talk with clients and patients in the future. That was the goal with your midterm as well, looking at Asha and her uh, life and how does the research that we've read apply to her. So a couple of useful terms that will come up. So ad libitum is referring to as much or as often as necessary or desired. Generally, desired is what I would read this research as. So usually it's referring to either however much sleep you want or however much food you want to eat. Okay. Hedonic eating just indicates that there is a pleasurable component to eating. So that's the hedonic piece. Free living is used in opposition to lab work. So when I bring someone into a lab and I measure their sleep, right, I know that that's going to affect how they sleep. Most of you have experienced this probably when you've slept in a bed that's not your own and how that is going to affect your sleep. Some of you maybe sleep better, but most of you probably sleep worse when you're sleeping in an unfamiliar environment. So we know that that's going to have an effect on the results that we get. So you'll see that come up in both articles, free living versus lab work. Whole body insulin sensitivity, I just wanted to talk about how there is a difference between liver insulin sensitivity, skeletal muscle insulin sensitivity, and then whole body insulin sensitivity. We've mainly focused on liver and skeletal muscle. Just know that this article mentions whole body, so they use intravenous measures as opposed to direct liver or skeletal muscle measures. The central circadian clock is mentioned. I don't think Chris will go this in depth on that, but I wanted to just really briefly talk about there's this main area of the brain called the suprachiasmatic nucleus. It is generally held responsible for circadian biology. So it responds particularly to light sensitivity and dictates when and how certain processes start in our body. You'll also see dim light conditions come up a lot. So because the SCN is particularly sensitive to light, often measurements are taken in dim light conditions or participants in a lab setting are confined to dim light conditions to prevent or rather sometimes control for the light component that we know is involved in these circadian processes. Homeostatic regulation of sleep comes up. I just wanted to give you an example for what that might be referring to. And Chris may talk about, or Dr. Depner, sorry, I should stop referring to him as Chris in here. Dr. Depner may talk about that a bit. Homeostatic regulation of sleep often refers to there is a buildup of adenosine in the brain while we're awake, and that adds what would be termed sleep pressure. So as adenosine builds up, we get more and more tired. You do see melatonin come into this as well. So, but melatonin isn't built up throughout the day. It usually starts to build up at a certain time in the evening, and then it tapers off before we wake, generally speaking. So in any case, all that homeostatic regulation of sleep is referring to is that there are certain biomarkers, certain chemicals like adenosine and melatonin that regulate our sleep and that regularly cycle up and down in response to our lifestyle behaviors and actions, or our exposure to sleep or light or food, etc. Just also to give you some context for how our behaviors can interrupt that process, caffeine is one of the ways that we can inhibit sleep pressure. So caffeine, and this is very superficial level. This is not my area. So Chris may go into more depth and he may provide more correct or more helpful information in this regard. But essentially caffeine is going in and it's attaching to the same receptors that adenosine would attached to. So if adenosine is attaching to a bunch of receptors, I'm going to feel tired. However, if I ingest a bunch of caffeine, caffeine's going to come in, block those receptors, meaning that I'm not going to be as sensitive to the adenosine buildup. Okay, as soon as the caffeine wears off, then I'm still building up adenosine, and now I might feel even more tired as soon as that caffeine wears off. Okay, so again, just how our behaviors may interrupt that homeostatic process. Couple of terms. Sleep latency refers to your time in bed before you've actually fallen asleep. So most of us know we get in bed, you don't fall asleep immediately. There's some time. So some people have really short sleep latency, some people have really long sleep latency, and some of that's related to our behaviors as well. Differentiating between sleep quantity and quality, right? Quantity would refer to the amount, quality would refer to the efficiency of sleep, essentially. So a couple of terms that are used, time in bed, right? Time in bed doesn't mean I'm asleep that whole time. So that could be that sleep latency piece of it. It could also be related to sleep efficiency. So sleep efficiency could be a couple of things, but we know that people wake up in the middle of the night regularly. Some people remember waking up, some people don't. But when we're looking at uh, electrochemical measures of what's happening in their brain, we can see, okay, they have woken up at this point, whether they remember waking up or not. So the number of times that you wake up, as well as the amount of time spent in different sleep stages, could both be referring to sleep efficiency. Social jet lag comes up in both articles. It is referring to sort of this mismatch between what society requires and what biology would encourage. So just a brief example, it's not perfect. We know people vary person to person in terms of sleep need and how they sleep, but let's just say in a perfect world, everyone falls asleep at 10 p.m. and wakes up at 6 a.m., right? Pretty typical what you might think of as a sleep window. We know that there's variability there. So some people are what we would call a late chronotype or an evening chronotype. And we know that adolescents in particular shift their biological clocks forward during a certain number of years while they're going through development. I don't know why, maybe Chris will go into it, but in any case, this happens. And so maybe normally I would be going to bed around 10 o'clock. Well, now I'm not going to bed until midnight, but school times often require me to still wake up at 6 a.m. So now I've just shortened my sleep window from 8 hours to 6 hours. That phenomenon is referred to as social jet lag. Per protocol analysis. This one I just wanted to bring up because I read in one of your midterms an intention to treat analysis. And so I wanted to compare these two things. So a per protocol analysis means that the participants that are analyzed are only participants who completed whatever the intervention was. So in a lot of your studies that you proposed for ASHA for the midterm, that would be, okay, if they were exercising 3 times a week, doing some type of aerobic activity for 30 minutes at a moderate intensity, right? Individuals had to have actually done that, and we had to have marked that they did that somehow. And then if they did that, then they were included in that statistical section of the analysis, right? As opposed to people who didn't complete the protocol. So that was your control group. Okay. So that's a per protocol analysis. Intention to treat analysis means that whatever group you were randomized into, so, right, most of you talked about randomization in your proposal of study design. So I had an intervention group and a control group, and I randomized people to that. Now, intention to treat says whether or not, so let's say you're in the control group and you actually decided to exercise, okay, on your own. You still stay in the control group for analysis. Additionally, if you were in the intervention group, in that aerobic exercise group, but you did not exercise, you are still analyzed as part of this aerobic intervention group. Okay. So intention to treat is trying to get more at real world analysis, right? I can prescribe someone something, but that doesn't mean they're going to do it. And so these are two different ways of analyzing. It can analyze per protocol, so we know the effect that the protocol can have, for example, whereas intention to treat might be more about what's the practical value of this intervention. Yes, we know it's effective because of the per protocol analysis, but with the intention to treat analysis, it wasn't effective. So maybe people can't do this on their own, therefore it's not very practical. Okay. The second study you'll read is a counterbalance crossover study design. So I just wanted to give you a bit, it can be a bit confusing when you first start reading the paper if you're not familiar with this. So all this says is that there are even numbers of people in groups and that they don't all follow the same order in terms of the intervention. So in this case, the intervention involved 3 days of baseline sleep, followed by either 5 days of short sleep or 5 days of 9 hour sleep opportunity. Okay. One of those two things came next. Whichever one came next, the other one was then that third piece. So if I had my baseline followed by 5 days of short sleep, then I had 5 days of 9 hour sleep opportunity. The other group, I had my baseline, then 9 hours of sleep opportunity for 5 days, and then 5 days of short sleep. Okay. So they're trying to get at the order in which somebody experiences interventions may may have an impact on the effect that the intervention has. So that's a counterbalance crossover study design, and you'll see that in your second article. And then the last thing I wanted to mention was shift work. Shift work just refers to people not working typical hours. So your typical hours would be like a 9 a.m. to 5 p.m., right? Shift work means that people are working outside of those hours. So firemen, pilots, lots of nurses, medical professionals in general, may be working outside of those hours on a regular basis. That would be considered shift work. Okay. Okay. That's all I have for you. Good luck with the articles this week. Let me know if you have questions. I think the narrative review is really interesting, should give you some really great background. And then also the that second paper, the Eckel 2015, should give you a good sense for how individual studies operate as opposed to these systematic reviews. So let me know if you have questions, and I will see you on Thursday.